Mitosis and meiosis have a lot in common. They're both cell division processes. They're both going to involve these same stages of prophase, prometaphase, metaphase, anaphase, and telophase, and they have a lot of the same things happening during those different stages. So we're going to focus on some key differences. The first is the outcome. Mitosis will make exact copies. So if you start with a cell that has 6 chromosomes, then you're going to make 2 daughter cells that also have 6 chromosomes. On the other hand, meiosis is going to produce 4 haploid daughter cells from the same diploid cell. So if you start with a mother cell that has 6 chromosomes, you're going to have daughter cells that have a random mix of 3 chromosomes. So again, mitosis will take a diploid cell and make 2 exact identical cells. Meiosis will take a diploid cell and make 4 unique haploid daughter cells. So in order to achieve those differences, there's going to be 3 key events in meiosis that, in particular in meiosis 1, that are very different. So we talked about these a little bit, and so I just want to re-emphasize them one more time. Synapsis and crossing over only occur in prophase 1 of meiosis 1. This is when the homologous chromosomes are going to physically touch each other, and while they're touching each other, they'll exchange matching pieces of information. So I didn't say this earlier, but I want to emphasize that the only way that this is a normal, healthy process is if it exchanges matching pieces. If they exchange unmatching pieces by accident, then that's going to create a mutation. Okay, so the normal process that they exchange matching pieces of genetic information. The other difference in meiosis 1 is that you'll see tetrads, or the term we're using more is bivalence. You'll see bivalence or homologous pairs on the metaphase plate. And again, in mitosis, you're only going to see a single chromosome. And then the third unique event to meiosis 1 is during anaphase 1, when you see separation of the homologous pairs rather than separation of the chromatids. In meiosis, the chromatids won't separate until anaphase 2 of meiosis. So again, sort of looking at a comparison here, mitosis starts with a diploid cell that has 6 chromosomes. Those 6 chromosomes line up individually on the metaphase plate. The sister chromatids separate, and you get 2 daughter cells that both each have 6 chromosomes. Meiosis will copy the DNA, and during meiosis 1, the bivalents form and crossover occurs. So you get that exchange of information. In metaphase 1, instead of seeing chromosomes lined up individually, you see chromosomes lined up in pairs, and that sets up those chromosomes to separate the homologous pairs. So at the end of meiosis 1, you have 2 cells that have one of this pair, but the chromatids are still together. It's not until meiosis 2 that the chromatids separate. And so that's how you get the 4 haploid daughter cells. So you go from 1 diploid cell to 2 haploid cells at the end of meiosis 1 to 4 haploid cells at the end of meiosis 2. The rest of the processes are very, very similar in meiosis and mitosis.